Hey guys, it's Medicosis Perfectionalis, where medicine makes perfect sense. Let's continue our biology playlist. We're talking genetics now. Mutations. You will learn about nucleotide mutations, and you will learn about chromosomal mutations, and all of their subtypes, including silent mutations, missense mutations, nonsense mutations, frame shift mutations, insertions, deletions, you name it. This is part of my biology playlist. Please watch these videos in order. Your body's building unit is the cell, which contains the nucleus, the brain of the cell. The nucleus contains DNA, which is made of sugar and nitrogenous bases and phosphate. If you add the sugar to the nitrogenous base, congratulations, you have a nucleoside. Add phosphate to them, congratulations, this is a nucleotide. When you add phosphate, it becomes nucleotide, as we have discussed before. So sugar plus nitrogenous base, nucleoside. Get that entire nucleoside and add a phosphate to it. So now we have three things. That's a nucleotide. Can this nucleotide have mutations? Yeah, we call them nucleotide mutations. This wonderful DNA is packed into chromosomes. Can we have chromosome mutations? Sure. Please pause and review the definitions in genetics that we discussed before. Now it's mutation times. Let's go. Mutation is a change in DNA sequence. Could be nucleotide mutation or chromosome mutation. Nucleotide mutations could be silent, missense, nonsense, insertion, or deletion. Chromosome mutation could also be deletion, duplication, inversion, insertion, translocation. Let's go back to nucleotide mutation. The silence, missense, nonsense are known as point mutation. Insertion and deletion are known as frame shift mutation. Nonsense is very important because it makes a stop codon, such as UGA, UAA, or UAG. How about UGG? Gigi is a female name in many countries, so have some respect. Gigi is not a stop codon. The stop codon always starts with a U and never contains Gigi. So it could be UGA, UAA, or UAG. A crazy mnemonic is, imagine that you're talking to the mRNA or the DNA and saying, here's your stop codon, therefore you go away. You are away. And you are gone. She gone, man. She gone. But UGG is not a stop codon. This is you go, girl. Don't let anybody stop you. You go. You are not a stop codon. Now here is the best slide in genetics since the invention of peanut butter. Nucleotide mutation, chromosome mutation. Nucleotide mutations could be point or frame shift. The point is silence, missense, or nonsense. Silence, what does that mean? No effect on the protein. Everything is hunky-dory. Thumbs up. Example, suppose that I had the following, TTA, and it was changed into TTG. TTA codes for what? Leucine amino acid. How about TTG? The same amino acid, leucine. So everything is the same, hunky-dory. Yeah, everything is perfect. Silence mutation. No one will notice it because it is silent. Next, missense mutation. Missense, look at the S and the S, substitution of one amino acid into another amino acid. For example, I used to have glutamate, but now I have valine instead of glutamate. Oh, this is substitution. Exactly. This is missense mutation. And this is what happens in sickle cell disease. Instead of having glutamate, now you have valine. The end result is that your red blood cell will be abnormal in shape. In other words, sickled instead of circular and biconcave. And like Dr. Thomas Sowell said, there are no solutions in life, only trade-offs. I have good news and bad news. Good news, malaria cannot live on this red blood cell. Oh, it cannot perform its life cycle there. Therefore, sickle cell is protective against malaria. Bad news, sickle cell disease is a disease. These sickled red blood cells can clog your vessels, causing all kinds of crises. And by the way, I have a video about sickle cell disease and sickle cell trait on my YouTube channel in my hematology playlist. How about nonsense? That's the stop codon. Example, I had UGG, it became UGA. This is a stop codon. Now it's gonna stop. That's nonsense mutation. Next, insertion or deletion. They can lead to frame shift mutation. What's a frame shift mutation? Consider this. Imagine that I used to be ATG, TTA, CCT. ATG gives you methionine. TTA gives you leucine. CCT gives you proline. And for some reason, this T was erased. Lost. Oh, because of deletion, for example. Now what? ATG will still give you methionine, but instead of having TTA, remember we dropped a T. So now I have T, A, C, which does not code for leucine. It codes for tyrosine. Oops. That's the frame shift because you're shifting the pattern, in this case by one nucleotide. Next, instead of having CCT, you only have C, T, and you borrow the next one, which is A. You shift the frame, and CTA will not give you proline. It will give you leucine. And so on and so forth down the road. By the way, this frame shift can encounter what? Rearrangement and a stop codon. And this can lead to nonsense mutation. So you can start with a frame shift that will make you stop, i.e. nonsense. As for deletion, you can delete a nucleotide, as we have seen here, or you can delete part of the chromosome, which is basically a sequence of DNA, like this. How about duplication? Well, I used to have one sequence, now I have 2x, double, or 3x, triple, or 4x, quadruple. All of this is duplication. Copied and copied and copied. How about inversion? The DNA sequence is reversed. I used to be purple, then green. Now I'm green followed by purple. That's inversion. How about insertion? Look at these two chromosomes. The first one had this red part, or red piece. The second one did not. The first one gave its red piece and inserted it into the second one. Oh, this is insertion. How about translocation? Translocation is exchanging, not just adding or giving. No, it's exchanging. It's give and take. Look at this. One of them had a green, the other had purple. All right, you take my green, I take my purple. You scratch my back, I scratch yours. This is translocation in a New Jersey accent. Here is sickle cell disease. I used to have glutamate, but now I have valine. Oh, this is missense mutation, which is part of point mutation of the nucleotide, not the chromosome. The end result is a cell that looks like this, sickled. Please pause and review. By the way, you can download all of these notes on my website. There is another disease called CML, chronic myeloid leukemia. It's one of the leukemias. It's cancer of the bone marrow. Here's chromosome 9 and here's chromosome 22. They will exchange. You scratch my back, I scratch yours. Chromosome 22 took a piece from the blue, which is 9, and chromosome 9 took a piece from chromosome 22. But on chromosome 22, there is fusion between BCR and ABL. We call this fusion gene. An example of reciprocal translocation. Scratch my back, I scratch yours. Got some reciprocity action going. And since this happened between chromosome 9 and chromosome 22, we call the translocation T for translocation 9 and 22. Please pause and review. If you want to be an excellent student, bring a blank piece of paper and draw everything here. Better if you can do it from memory. If you like this video, you will enjoy my renal physiology course on my website, medicosisperfectsnellis.com, or my brand new surgery high yields course, also on my website. Your purchase and download will give you a lifetime access. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my courses. Be safe, stay happy, study hard. This is Medicosis Perfects Nellus, where medicine makes perfect sense.